A grief that lasts long enough can leave a physical mark on the brain. Not a feeling, not a metaphor, a measurable loss of tissue you can scan. In brain scans of depressed patients, one region appears visibly smaller. Studies report the hippocampus shrinking by up to 10% in chronic depression. The hippocampus is your memory center, and depression seems to eat into it. For centuries, we thought the mind and the brain lived in separate worlds. Then imaging machines opened a window, and the worlds collapsed into one. The brain is the only organ that can study itself. In the late 1990s, psychiatrists began scanning the brains of depressed patients. Sheline studied women with recurrent major depression using high-resolution MRI scans. The longer the depression, the smaller the hippocampus. A dose-response pattern emerged from the data. Her 1996 study found roughly 10% volume loss in the hippocampus. The damage seemed to track total days spent untreated and depressed. This was the first hint that mood disorders leave fingerprints in tissue. The leading suspect was a hormone your body makes under stress. Cortisol floods the bloodstream during threat, fear, and prolonged pressure. In short bursts, cortisol saves your life. In chronic excess, it may corrode neurons. What protects you in a crisis can poison you in a marathon. The hippocampus is packed with receptors that respond to cortisol. That makes it exquisitely sensitive and exquisitely vulnerable to stress hormones. Sapolsky spent decades studying stress in wild baboons and lab rats. Sustained cortisol shrank dendrites, the branching arms. Of neurons, cells did not die, they retreated. Neurons pulled back their connections, like trees losing branches in winter. In stressed animals, dendritic branches in the hippocampus dropped sharply. Fewer branches means fewer connections, and a brain that communicates less. For decades, scientists believed the adult brain grew no new neurons. Then they discovered the hippocampus makes fresh neurons throughout life. Duman proposed that depression slows this birth of new brain cells. This process is called neurogenesis. Stress and depression appear to suppress it. Less neurogenesis, fewer new cells, and a hippocampus that slowly thins. Antidepressants take weeks to work, but they raise serotonin in hours. That delay puzzled researchers for years. Why the long wait? Duman's lab found antidepressants boost neurogenesis over weeks. The timeline matched when patients felt better. Maybe the drugs work by helping the brain grow itself back. In rodent studies, blocking neurogenesis erased the antidepressant effect entirely. Recovery may be the brain rebuilding what stress tore down. A protein called BDNF acts like fertilizer for neurons. Depression lowers BDNF levels in the brain. Treatment and exercise raise them again. Exercise turned out to be one of the strongest BDNF boosters known. The shrinking is not permanent damage. It is reversible plasticity. The brain did not break. It adapted, then waited to be rebuilt. Depression is not weakness. It is a brain under biological siege. The fatigue, the fog, the lost memories, these have a physical basis. Effective treatment may restore lost volume over time. Studies suggest the hippocampus can recover. That is why early treatment matters so deeply for the brain. If you are struggling, this is medical, not moral. Seek a professional. Next time someone says depression is all in your head, agree. It is in your head, literally, biologically, and reversibly.